Hello, everyone, and welcome to today's webinar titled, Will We Have a Vaccine for COVID-19? 5 Reasons for Optimism. This webinar is part of an ongoing coronavirus virtual webinar series. I'm Susie Valdez of Labroots, and I'll be your moderator for today's event. Today's educational web seminar is presented by Labroots. So let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you'd like during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing this presentation, simply click on the Ask a Question box located on the far left of your screen. As a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits tab located at the top right corner of your presentation window and follow the process for obtaining those credits. Without further ado, I now present today's speaker, Dr. Rob Kozak, clinical microbiologist, Sunnybrook Health Sciences Center, associate professor at the Department of Laboratory Medicine and Pathobiology at University of Toronto. For a complete biography of our speaker, please visit the Biography tab at the top of your screen. Dr. Kozak, you may now begin your presentation. Welcome, sir.
Thank you so much, Dr. Valdez, for the kind introduction, and thank you to my online audience, everyone for coming. It's a pleasure to be able to talk to you today a little bit about vaccines, and I hope by the end of this you'll leave with the same optimism that I have. So just in general, today we're going to talk about several of the vaccine candidates that are in current development right now. As well, I'm going to talk a little bit about some of the lessons that I think we can learn from SARS and MERS. There's going to be a few considerations which I think can also be not just challenges but opportunities for the scientific community moving forward. And of course, I'll leave you with my 5 reasons for optimism at the end. I'd just also like to say that I have no relevant conflicts of interest or financial disclosures. So I don't think anyone would agree that we really do need a vaccine for COVID-19. This is a sign sent from a colleague of mine, which is quite humorous, but also kind of highlights that even though we have a lot of good public health measures in place, such as social distancing and the use of masks, and that has been really useful in certain areas at flattening the curve, in order to get things back to some sort of normal, we are going to need to have herd immunity within the general population, and that will likely require a vaccine. So if you're a real vaccine, if you're really passionate about vaccines like I am, you can actually follow on the New York Times has a real-time vaccine tracker, although the pace of vaccine research, I don't know how useful a real-time tracker truly is. But I think it's interesting because it really highlights that there are quite a number of different vaccines that you can see there in various stages. So there's just over 100 in the preclinical stage, and then there's a handful that are in phase 1 and phase 2, 2 that are currently in phase 3. And to date, we don't have any that are approved. And this isn't surprising because if you think about all the steps that really take to get a vaccine from concept all the way to putting it into the into people to confer protection, it's quite a long road. And usually this is a process that can take potentially decades to accomplish. So even after the clinical phase, after you've done all the scientific work, which we'll talk about in a moment, there are a number of other steps that are really important, but are often not really considered, by frontline researchers. So first off, there's the whole good manufacturing process. So you may have an excellent vaccine, but you have to be able to manufacture it properly and under very stringent conditions if you're going to then use it for clinical trials. Of course, there are the different phases of clinical trials, which I'll go into in just a moment. But assuming that your vaccine makes it all the way through and is successful in each of the different phases, at that point, you're still not home free because There is the requirement for licensure and approval by the various regulatory bodies. And of course, something that often is not considered by a lot of researchers is the need for being able to scale up production. So especially for something that's a pandemic, you're going to need to create likely billions of doses of this. So the production can be a bit of a bottleneck. Then even if you do have the production in place, there's also the rate limiting step of administration. So it's not like you can just automatically give out the vaccine to billions of people. You have to figure out how to administer it, how to distribute it, and then with every vaccine, it takes time for following the vaccination in order to generate immunity. So it's if we're able to get one within 18 months, that will be at just a record pace. But you can see now why it often takes quite a long time. And if we go back even just to the preclinical side of vaccine development, which is probably what a lot of people think about when you think about research being done in the lab. There are some things that you need there. There are some challenges to overcome. So first off, you need to select an antigen, and then you also need a candidate vaccine. So you need a vector or some sort of system that you can use to deliver that vaccine into either animals and then later on into people. You also need the pathogen, so you need to have the virus isolated in order to do the next step, which is the animal challenge models. So you also need a good animal model of the disease in order to properly evaluate whether or not a vaccine is efficacious. And probably one of the most critical things, you need the funding and resources in order to support this. These are long studies, and money is certainly required. And once you get through all this, that's when you can actually move into the clinical trials. So probably a lot of people in the audience are familiar with the different phases of clinical trials, but I'll just give a really quick overview of what we mean by each of them, just so that we're all on the same page. So when we talk about phase 1 clinical trials, these are the trials where you have generally a small number of individuals who are given the vaccine, and what you're really looking for here is the safety profile and identifying the optimal dosage of your vaccine candidate. If you're successful here, you then move on to the second phase, phase 2, and these are the larger trials that are used to generate immunogenicity. They often involve a diverse group or diverse segments of the population in order to see that the immunogenicity is constant in all groups. So it might include the elderly, or it might include children, depending on your vaccine. And then if you're successful there, you move on to the next phase, which is the phase 3. This is the large-scale trial that evaluates the efficacy of the vaccine. So the good news is that often these phases can be combined. So you could have, for example, a phase 1-2, or as we'll see in a moment, some of them are in phase 2-3. So that can sometimes save time, but vaccines can really fail at any stage, and not just because they may fail for scientific reasons, such as they really fail to generate immunogenicity, sufficient immune response, or medical reasons like they have too many adverse effects, but often factors that can cause it to fail that we often don't consider. So things like you may not be able to enroll enough people in order to achieve sufficient power to evaluate your vaccine, or you may run out of support and funding in order to move to the next phase, which is actually what we saw with the some of the vaccine trials for SARS-CoV-1 back in 2004, 2005, where a number of vaccines made it into phase 1 trial and then didn't progress further from there. So one of the other things I said we need in the preclinical work is obviously the virus. So there have been a number of groups that have isolated the virus all around the world, and this is excellent because it's now available to researchers everywhere for performing these studies. So I'm sure everybody is well familiar by this point in the conference what the virus is. But again, just so we're all on the same page, so when I'm talking about SARS-CoV-2, which is the etiological agent of the disease COVID-19. It is a coronavirus, part of the beta coronavirus genus. Like other coronaviruses, a positive-sense single-strand RNA genome. It's enveloped. Like SARS-1, it uses the ACE2 receptor. And one of the critical proteins that I'm going to talk a lot about today is the spike glycoprotein, which you can see the picture in the corner, the yellow proteins that are all around the edge of the virus, or they can also be seen in the electron micrograph just at the bottom of the page. So these proteins are what's used by the virus to bind to the ACE2 receptor and enter the cell They also are the reason that the virus got its name, corona meaning crown, because these proteins give it sort of a crown-like appearance on the electron micrograph. So that's good. We have access to the virus. We also have a number of animal models, which, as I said, is very important for a lot of the preclinical work. We were fortunate that a lot of the knowledge that we had from SARS and MERS, we can kind of apply to COVID-19, which I think really sort of expedited the process and really made development of animal models go a lot faster. So from SARS and MERS, we knew that non-human primates and ferrets were both excellent models for performing various pathology studies and also for vaccines and therapeutics. The ACE2 transgenic mice we know was used for SARS. And since then, we've also determined that Syrian hamsters are also another animal model for COVID-19, and they're also able to mimic transmission, as do ferrets. So each of these models, they have their advantages and disadvantages. Obviously, smaller models such as mice and ferrets and hamsters are easier to cage and are a little bit less costly than non-human primates. But non-human primates are advantageous because they are more closely resemble what we see in humans. And basically, with a lot of the development of animal models involves determining the best way to challenge the animal to mimic as close to possible what we see, the disease we see in humans. So for this, what we do is an intranasal challenge with the virus, and there is value to determining what is the optimal dose. And then following the intranasal challenge, what you do is you monitor these animals for viremia, you look for viral shedding in nasal swabs, and you do evaluation, clinical scoring to look for disease. And at various time points, you may euthanize the animal in order to look at the viral burden in organs as well as histopathology. Ideally, what you're hoping for when you have an animal model is you want a lethal model. And why we want that is because it's a lot easier to see whether your vaccine or if you were testing a therapy, whether or not it's really having an effect. If you have a lethal model and your vaccine prevents animal death, that is advantageous. Unfortunately, the models that people have developed so far are not uniformly lethal. Some of the animal models, there is a point where some of the animals will succumb to the illness. They're not uniformly lethal, which adds challenges when we're doing vaccine studies. So recently, there was a paper published in Science, which was excellent. It really highlighted that non-human primates are a really, really good model of COVID-19, and they really mimic what we see in humans. So I've just highlighted a couple of key figures from the paper. I encourage you all to take a look at it. This group actually took both young and aged non-human primates and infected them with SARS-CoV-2, and actually showed that the disease phenotype is similar to what we see in humans. So generally we see in humans, older individuals, we see a much more severe disease with a higher viral load. And we see the same here. So you can see in those figures, the black bar represents the aged animals, which is high, and you see higher viral loads and more dissemination into the organs than you do in the younger animals. And what was also interesting is they actually did a head-to-head comparison of both SARS-CoV-2, SARS-1, and MERS. So they did a full-on pathology study to compare the disease course in all these using all these different viruses in these non-human primates, and really demonstrated that the non-human primate really represents is an excellent model of what we see going forward in human disease. And so this is very encouraging because, as I said, non-human primates are really our gold standard model for looking at vaccine efficacy. And so having one that accurately represents the disease that we see with COVID-19 is very important. Okay, so what have we learned from SARS and MERS that can potentially help us get a vaccine sooner with COVID-19? So what we saw with both SARS and MERS is that there were actually a whole range of different vaccine candidates and types of vaccines that were were investigated. So there was no real one that sort of stood out saying, oh, this is the exact type of vector, or the exact type of vaccine that works and nothing else does. So this is encouraging. It means that there's lots of different options on the table. And the different vaccine types that were examined they kind of run the spectrum. So on one end, you've got your live attenuated and then your inactivated virus. So these are using the actual virus and either deleting part of it or inactivating it through chemical treatment. And these have their advantages, that they are often what is closer to natural infection in the case of live attenuated. And they're easier to, they're quite easy to develop. You can develop them quickly using an inactivated vaccine, but both of them have safety concerns. Now, if you move further down the spectrum into using a viral vector, so another virus where you get it to express the one of the antigens from either SARS or MERS, these are a little safer and they do generate a good immune response, but they may not work in all segments of the population. So people that are immunocompromised may not be able to tolerate them. And also, like any live viral vector, there are difficulties in production. The ones that are probably the easiest to produce are the subunit vaccines, so a protein-based vaccine or a DNA vaccine. These were shown to be easy to produce. They obviously have very favorable safety profiles. The problem is that they may not always be immunogenic. You have to determine what's the ideal antigen, and so as a result, they may have the requirement for multiple doses. But generally, what we take from this is that all forms of all the different types of vaccines have shown varying degrees of efficacy and were investigated for SARS and MERS. So there's lots of options on the table. And I should also state, for just in case you're not familiar with vaccine studies, generally when we're looking at vaccine efficacy, there's really kind of three things that we're kind of focused on. So we want to make sure that the vaccine will generate virus-specific antibodies and T cells and a high titer of antibodies. We also want to make sure those antibodies are neutralizing, because although it hasn't been determined all the correlates of protection for SARS-CoV-2, generally speaking, neutralizing antibodies are associated with protection in many viral illnesses. And of course, probably the most important thing, we want to make sure that the animals that are vaccinated survive. And if we don't have a lethal model of infection, we at least want to see that the vaccinated animals have a more moderate disease, which can mean lower viral burdens, less clinical symptoms. We want to see that they're doing better than the animals that get the sham vaccine. So now, in terms of selecting antigens, again, we can learn a lot from what we saw with SARS and MERS, and this has been applied, I think, by a lot of the vaccines for COVID-19. Rather than testing out and trying all the different proteins that are present in the virus, the spike protein, which encodes that glycoprotein that I showed you a few slides ago, that sort of coats the outside of the virus, this is really a good choice for an antigen. So the spike protein, or what I would just refer to as the S antigen, it's the one that binds to the host receptor and allows the virus to get in. So obviously, developing antibodies against this will help neutralize the virus from being able to enter the cell. And there were a number of studies that were done with SARS that showed if you took antibodies against the SARS protein from survivors and passively transferred them into small animal models, you also conferred protection. But a really elegant study was actually this one done in PNAS in 2004. And so what this group did, this table's a little bit busy, so I'll just walk you through it. What basically they did is they took parainfluenza virus and through a little bit of genetic engineering, got the parainfluenza virus to express various proteins from SARS, so either the S protein or the E protein or the nucleocapsid protein, and then they used these to vaccinate animals and looked at whether these vaccines conferred protection. So in the very last column, what you can see where it says % with detectable SARS-CoV in the lungs, the only ones where there was no detectable virus was in the mice that had received the vaccines containing the S protein. So none of the other proteins. None of the other vaccines expressing the other proteins really induce protective immunity. So this really shows us that the S antigen is probably the best bang for our buck. And we also see similar studies from what's been shown with MERS. So this is from a couple of recent studies where there was a group that was studying the DNA vaccine that expresses the S gene for MERS. MERS. So again, the spike glycoprotein from MERS. And what they found is that in non-human primates, the vaccination induced high titers of antibodies and neutralizing antibodies against MERS-CoV, and that these non-human primates, that I didn't have space to show the data, but vaccination with this plasmid containing the S gene conferred protection. They then went on to do phase 1 studies and showed that in healthy individuals that the DNA vaccine that expressed the S antigen for MERS was immunogenic and had very minor side effects. So this is really helpful because this really shows us that this could be a potential strategy for a COVID-19 vaccine. And indeed, there's a couple companies, including Inovio, which is actually pursuing this with a DNA vaccine of their own against COVID-19. This is again another busy table from their Lancet paper, but it's just to highlight that they tried different dosages, which is what you're usually trying to do in a phase 1 study, and they showed that there were very few adverse effects, even if you used a high dose of the vaccine. And that's important too, because it's not just immunogenicity. If you have a number of adverse effects, not only is that has the potential to harm the individual who's receiving the vaccine, but it can also be a bit of a challenge when you're trying to roll out a large-scale vaccination program. Okay, let's move into some of the current candidates that are on the market hoping to one day be on the market for COVID-19. So as you remember, as I showed at the beginning, there's over 100 candidates, so I obviously don't have enough time to go through all of them. So I just highlighted a couple here that have progressed into clinical trials. So you see that, again, there's a spectrum of different vaccines that are being investigated, including a subunit vaccine from using the S protein from Novavax. Both CanSino Biologics and University of Oxford are using adenovirus vaccines. Inovio, that I alluded to, is using a DNA vaccine, and Moderna, which is probably a lot of people have heard about, is using an RNA-based vaccine that encoding the S antigen. So I think the thing that's interesting is that, and again, really builds on the knowledge that we gained from SARS and MERS, is that all the vaccine candidates are using the antigen, just highlighting what a promising antigen it is. The status of these ones tends to differ. It's unfortunate. There's not a lot of scientific literature on the findings from some of the preclinical work and the phase 1 study. So hopefully that'll become available soon, so that the scientific community can start evaluating these different vaccines. But we'll just have to wait and see. So I'm going to start with one of the ones which is the most furthest advanced. So it's what's called the ChAdOx1, which is stands for the chimp adenovirus from Oxford, and this is the one that the chimp adenovirus that I showed you in the previous table, which has been genetically modified to express the S antigen. And again, so it has moved past phase 1 trial. It's in phase 2 and 3, and unfortunately, I wasn't able to find any published literature on the phase 1. Hopefully, that'll be soon. So instead, I'm going to present some of the preclinical work that was done in animals, and I think it's quite promising. But again, I should caution you that the data I'm showing you is from a preprint, so it hasn't technically gone through a formal scientific review. So what this group has shown is that when you vaccinate either mice or so what they did is they vaccinated mice and non-human primates, and they were looking for both immunogenicity, and actually in non-human primates they were going to do challenge studies. So for immunogenicity, you're looking for the trifecta. You're looking for virus-specific antibodies, a nice high titer, specifically neutralizing antibodies as well, and virus-specific T cells. For the challenge experiments, they vaccinated the non-human primates, and then they challenged them with a relatively high dose of SARS-CoV-2. So what you can see here, this is their experimental design. So all the bars in red represent the data from animals which have received the chimp adenovirus, which has the S antigen for COVID-19, and then the blue is the one. These are from animals that received chimp adenovirus expressing the GFP protein. So the green fluorescence protein that's not from a virus, and it's essentially it's your control group, it's your sham vaccine. So what they did is 4 weeks before challenge, they vaccinated their animals. They drew blood at 2 weeks later, and then 2 weeks following to look at the immune response. Then they did a challenge and examined the animals up until day 7, when they did a necropsy to look at pathology and viral load. So from this, what you can see is we're getting the trifecta. We're getting in the vaccinated animals. We're seeing a good, strong immune response. There's high titers of both antibodies and specifically virus neutralizing antibodies, and good T cell response seen both in the non-human primates and in the BALB/c mice that they used. But obviously the question is, immune response is great, but does it actually confer protection? So again, we're challenged by the fact that this is a non-lethal model. So we don't know what the animals don't die from this disease. So all that we can look at is the viral burden and the clinical scores. Excuse me. What we saw is that the vaccinated animals actually had lower clinical scores, suggesting a more moderate disease compared to the animals that received the sham vaccination. Also, in the sham vaccinated group, 2 out of 3 developed pneumonia, whereas in the vaccinated group, none of the animals developed pneumonia. And then when they performed necropsy 7 days after infection, there was lower viral loads in the animals that received the chimp adenovirus that had expressing the S antigen. What was interesting, though, and I unfortunately didn't have space to put it in here, but if you look in the paper, at day 1 and day 3, they noticed that there was no difference in the viral loads for the nasal swab. So this suggests that perhaps this vaccine, while conferring protection against severe disease, may not actually confer protection against transmission, which is a bit disconcerting. But obviously, we need further studies to really investigate this. So overall, I think that this vaccine it looks incredibly promising, at least based on the animal data. They were able to confer protection in non-human primates with only a single dose of vaccine. And that's really important because, if you're going to roll something out into the general population, you ideally want a vaccine where you won't have to have people come back repeatedly for multiple doses. Another appealing aspect of this particular vaccine is that pre-existing immunity against the adenovirus is not likely, because this is a chimp adenovirus. When you use human adenoviruses, there's always the concern of pre-existing immunity, which could limit vaccine efficacy. But this, as I said, a little bit less of a concern. From looking on clinicaltrials.gov, they have completed a trial in over 500 adults. And so hopefully we'll see the data soon on that. They've also the University of Oxford has also struck a partnership with AstraZeneca, which will be useful, hopefully, for production of the vaccine. And right now, phase 2/3 trials underway. With a little bit of luck, we may see some data by the end of the year. So the next one I'm going to talk about is one that's completed phase 1 trial recently. So not as far advanced as the chimp adenovirus. This is one by CanSino Biologics, which is using an adeno5 vector, which is again expressing the S antigen from SARS-CoV-2. So in their recently completed phase 1 trial, again just to highlight a little bit of background, so adenovirus 5 is it's a human adenovirus, so it's one that people could get exposed to, may have been exposed to previously. It causes respiratory infections, so there is a likelihood of pre-existing immunity in populations. But so what they did is this is a replication defective adenovirus 5, but they have genetically modified to express the S antigen from SARS-CoV-2. And this was a dose escalation study, single center, open label, non-randomized. And they had just under 200 volunteers who kind of spanned a whole age range. They did note that up to 10%, or depending on the group, the vaccine group, more than 10% of their participants actually did have pre-existing immunity to the adenovirus 5 vector. But they did account for that. When they looked at the immune response, they did find that their vaccine does generate a very robust immune response, so good antibody titers and good T cell response. And they actually did break it down and showed that even in the group that had significant titers against adenovirus 5, which you would suspect would make which would actually decrease the immunogenicity of the vaccine, because your pre-existing immunity would prevent your immune system from potentially taking in the virus. They actually saw that you actually get fairly robust response comparable to the group that didn't have pre-existing immunity, which is encouraging. But where we, I think, have to pause a little bit is in terms of when they presented the table showing the adverse effects. So I'll just draw your attention to the fact that over 75% of participants in each category reported adverse effects, and some of these adverse effects could actually be pretty serious. So fever, pain, headache, fatigue, this is not. It is not uncommon in a lot of vaccine studies to see to have side effects, pain particularly, often at the site of injection. But where I think this is, it could be potentially problematic is some of these adverse effects are quite serious, and you can imagine they can, things like fever and fatigue can actually mimic some of the symptoms you see with COVID-19, which that in combination with just the high likelihood of a participant having an adverse effect, this could be a problem for getting the population as a whole to want to take this, to want to sort of uptake this vaccine. So it's something that I think we really need to consider. It's also important to remember that previous vaccines using the adenovirus 5 vector have had problems, such as the well-known one in 2007 with the HIV vaccine, where they actually showed that because of the pre-existing immunity, or what they believed to be pre-existing immunity, caused an increase in inflammation, which actually increased the likelihood of virus acquisition. So what we don't know, because this was obviously a safety and tolerability, safety and dosage trial, is we're not sure whether pre-existing immunity to adenovirus 5 will exacerbate the disease in the context of COVID-19. And it's, as I said, the high percentage of people who had adverse effects is, again, a little disconcerting, because when you compare it to the recent Lancet ID study that I spoke about earlier, using a DNA vaccine against MERS, DNA vaccine for MERS, this was also a phase 1 study, but showed that the individuals reported far fewer adverse effects. So perhaps it is important to really find the right vector for delivering your antigen. So a lot of these questions we can't answer until we do further trials and further studies. A phase 2 trial is underway, so all we can do is watch and hope that the scientific community is looking into some of these questions. So now I'll talk a little bit about a Canadian preclinical vaccine candidate that I'm actually fortunate to be working on. So this is part of a research consortium amongst researchers at the University of Toronto, Sunnybrook Health Sciences Centre, where I'm at, and the Université Laval in Quebec. And so what we're actually doing is we're going to be comparing a number of different vaccine candidates, including a DNA vaccine expressing the S antigen, virus-like particles, and a protein subunit vaccine. And we'll be determining which ones in preclinical studies are the most efficacious. We have started some work. We've done immunogenicity studies, starting with the DNA vaccine. That began in April, and we're just working on, we're about to start our animal challenge experiments. To give you a little bit of background, so DNA vaccines are basically, they're basically circles of DNA. If anyone's ever worked with bacteria, you're probably very familiar with plasmids. So this is a commercial system that you can order from Invitrogen, but the idea is the same. So the one that we designed is called PIDV, which is basically a modified and improved version of the PVAX one. We've done a number of manipulations to it in order to increase antigen expression, but the common theme for DNA vaccines is basically what you have. You have this circle of DNA, and you get to insert your favorite antigen here. In our case, we've used the S antigen from SARS-CoV-2, and there's a selectable marker and a promoter system so that when this vaccine is delivered into cells, it will express the antigen in order to excite the immune system. So we just recently completed the immunogenicity studies in BALB/c mice. So this is data hot off the presses, the hard work of one of the postdocs, Dr. George Babuadze, was able to provide this data to me earlier this week. So it's looking quite promising. He's been investigating our DNA vaccine with three different versions of the SARS-CoV-2 S antigen. And he's shown in cell lines that it gets we get really robust antigen expression. And then in BALB/c mice, we're able to generate pretty significant titers of both antibodies against SARS-CoV-2, as well as a robust T cell response. So the next step is, now that we've showed that this vaccine is immunogenic, we'll be moving forward into actual vaccine challenge studies. So stay tuned. Okay, so there's a couple of things that I think that we need to consider for all vaccines moving forward, and I think these are not just challenges but also potential opportunities for the scientific community. So one of the things that is discussed and brought up quite frequently is this idea of antibody-dependent enhancement. So if you look at the figure just to the side of your screen right here, under ideal circumstances, if you have antibodies against a virus, so let's say against SARS-CoV-2, the antibodies will bind to the virus and neutralize it in order to prevent the virus from binding to the receptor and entering the cells. And that's what we're hoping for with a good vaccine. Now, the concern is something called antibody-dependent enhancement, whereby the antibody will not bind quite correctly, but will bind to the FcR receptor will bind to the cell and then facilitate virus uptake, or the other one, which is immune-mediated enhancement, which is kind of like a different flavor of antibody-dependent enhancement, whereby the antibodies actually cause not only virus uptake, but also the generation of a pro-inflammatory immune response. And this is so this concept of ADE is actually quite familiar for a lot of other viruses, including there have been reports of it for influenza. But probably the most well known is actually with dengue virus, and we saw this with this was a concern with the dengue vaccine 2 years ago. But in terms for coronaviruses, this phenomenon has been observed when we do in vitro studies. So antibodies against the S protein have been shown to enhance coronavirus uptake for different coronavirus species, not just SARS, at least in cell culture. And also, when people have done these sorts of studies using animals, again, they have noticed what this sort of increase in pathology. So this is a recent paper published just back in 2019 in JCI. So what was done is rhesus macaques were vaccinated either with the vaccinia virus expressing the S antigen from first SARS, from SARS-CoV-1, or they were just given vaccinia virus. So that was the control vector. And when these animals were vaccinated, then subsequently challenged with SARS-1, and then necropsied after infection, they reported that the vaccinated animals actually had greater lung histopathology compared to the animals that had received the vaccine that didn't have the S antigen. And they also did studies where they said, well, if we give antibodies that are specific for this, for the S antigen, if we give it to animals, what do we see in terms of pathology following infection? And again, higher histopathology scores indicating there's more inflammation and more pathology in the lungs. So I should highlight this was done with SARS-1, not with SARS-CoV-2. But I think so that is quite compelling data, and it's certainly interesting and worthy of further studies. But what's interesting is when you go through some of the fine details of this paper, they did do radiography. So they did like a chest X-ray on the animals throughout the course of the study, but they didn't report whether or not the radiography was worse in the vaccinated, or so the animals that received the SARS-CoV vaccine compared to the ones that received the sham vaccine, or if it was just even worse than animals that were unvaccinated and were just infected with SARS-CoV-1, which they didn't was not a group that was included as part of their study. They also didn't comment on whether the clinical signs and symptoms were actually worse. So did one group of animals have more lethargy, more shortness of breath, decreased appetite, which are all things that are easy to measure when you're doing an animal study with non-human primates. So other than the histopathology, they didn't present any evidence showing that the non-human primates actually did worse in the end. So it really makes you wonder, did the animals even notice that they had this antibody-dependent enhancement? The only way we can know that for certain is whether we actually have to do these studies in non-human primates, obviously with our vaccines against COVID-19, and we need to do a longer follow-up study. So we need to, once the animals recover, we'll need to follow them and really examine to make sure that there isn't any additional pathology. It may even require a subsequent second challenge just to make sure that to determine whether or not there is any antibody dependent enhancement. These are all very important things to determine, that we want to know this before we start licensing and approving vaccines. We want to make sure there aren't any unintended side effects. And I think this is a great opportunity for the scientific community to investigate this and answer some of these questions. So other things that we need to consider is, obviously, if we have a promising candidate, we'll need to scale up production. So we'll need to be able to develop many doses of the vaccine in order to vaccinate subsequent or a large enough component of the population in order to confer immunity. So it's actually quite encouraging that there's so many candidates. In fact, if you ask me, I'm hoping that there isn't just one vaccine that works. I'm hoping there's 3 or 4 or half a dozen, because this will really alleviate a lot of the production problems. If you have 6 vaccines that are all working equally well, then that's 6 different companies that can be producing it in 6 different sites or in 6 different places, and this will really help get the population vaccination level up faster. We also need to understand a little bit whether or not we can develop a vaccine which will not just protect against severe disease, but may also protect against transmission. As I showed some of the preliminary data from the chimp adeno vaccine, it's suggesting that maybe it doesn't prevent against transmission, but the only way to know that is with good solid animal studies, and of course doing follow-up studies during your various phases of clinical trials. Also, we need to figure out now, what level of protection are we willing to tolerate? So it would be ideal if we had a vaccine that was 100% effective and protects people. I don't think anyone would argue with that. But when it comes to deciding, are we going to be able to use a vaccine that's only 60% protective? Probably. But what if our top candidate's only 40% protective? So these are all important questions that we need to consider now, and of course, and once we know that level of protection, that will also help us develop what would be the optimal vaccination strategy. It's unlikely that we're going to be able to generate billions of doses very quickly, so we'll have to determine. I don't know how feasible it would be to vaccinate 5 billion people. I don't make vaccines for a living. But even if with optimal production levels, and some companies are saying they will be able to produce millions, if not billions of doses, we may have to determine what will be the priority population. So potentially, the elderly, the immunocompromised, healthcare providers. A vaccine strategy needs to be developed in advance so that once we have that ready to go, that we don't lose any time. So I'm a glasses half full kind of guy. So I hope that I've sort of conveyed a note of optimism, but I think to summarize, I think there's 5 reasons that I'm very optimistic that we're going to have a really good vaccine against COVID-19. The first one being, we are only 5 months after identifying this virus. I know it seems like it's 3 or 4 times that, but it's been 5 months since we identified SARS-CoV-2, and already we have more than 100 candidates that are in either preclinical or various stages of clinical trials. So that is incredibly encouraging. We've got all the tools that we need to do a lot of these studies. We've got good animal models. We've got the virus isolated. We've got what we need to really push things forward. We've also saved a lot of time and learned a lot, I think, from lessons from SARS and MERS, and I think that's why we are so far ahead compared to how we would be had we not learned anything from SARS and MERS. We have multiple candidates moving forward, which again, I think is really important, because then we don't have reliance on a single winner. In fact, as I keep going back to my hope is that by the end of the year, we have maybe 4 or 5 candidates that are either about to be licensed or in phase 3 trials. And I think that seeing the speed that and the sort of the will of the scientific and medical communities, I think that there is an excellent chance that we will see some phase 3 clinical trial data by the end of the year. So with that, I'd just like to thank some of my colleagues who've been involved in our own little piece of the vaccine development for our vaccine consortium. So that's my colleagues here at Brook Health Sciences Center, Dr. Samira Mubaraka and her lab, and my colleagues at the Université Laval, Dr. Gary Kopinger, Dr. George Babouadze, who has been just instrumental in pushing a lot of us forward, and Dr. Gary Wong. And with that, I would be more than happy to take any questions.
And thank you, Dr. Kozak, for your time today and for your important research. And before we go, I'd like to thank the audience for joining us today. And questions submitted today and during the on-demand period will be addressed by the speaker via contact information you provided at the time of registration. And this webcast can be viewed on demand through the end of the year 2020. Labroots will alert you via email when it's available for replay. And we encourage you to share this email with your colleagues who may have missed today's live event. Until next time, take care and bye-bye.